Hello and welcome to the Vodcast about cell division. First of all, we're going to have a look at a double-stranded chromosome. In a double-stranded chromosome, we've got two identical sister chromatids. The telomeres are the ends of the sister chromatids. In the center, there is a centromere. The centromere holds both the sister chromatids together and also allows the chromosome to attach to the spindle fibers. The position of the centromere can vary. If it's in the center, we call it metacentric. If it's off-center, we call it submetacentric. If it's near the end, it's called acrocentric. And if it is at the very end, we call it telocentric. But most of the time, we just think about the double-stranded chromosome as being metacentric. Cell division only occurs after interphase has occurred itself. And in interphase, in the S stage, the single-stranded chromosomes become double-stranded. And in a double-stranded chromosome, each half is called a sister chromatid. Somatic cells undergo mitosis. Mitosis has the stages prophase, metaphase, anaphase, telophase, and it creates daughter cells. In prophase, the nuclear membrane breaks down and the DNA becomes visible as the DNA condenses. In prophase, the centrioles also start to make the spindle fibers. In metaphase, the chromosomes attach to the spindle fibers by their centromeres at the equator. In anaphase, the centrioles start to wind in the spindle fibers. The double-stranded chromosome splits in the middle at the centromere, and the chromatids separate. One sister chromatid moves to each opposite pole. In telophase, the nuclear membrane reforms around the collected chromatids. A cleavage furrow then forms, and cytokinesis occurs. In cytokinesis, two daughter cells are made. The two daughter cells created are identical to both each other and the parent cell. They are in fact clones. There's no variation. This form of cell division is perfectly suited to a constant environment. Let's look at cytokinesis in a little bit more detail. In the animal cell, we have the cleavage of the cytoplasm, and it happens at the cleavage furrow. The cells pinch off and two daughter cells are formed. In a plant cell, though, the cytoplasm divides and a cell plate forms from cellulose. Okay, now let's look at meiosis. In prophase 1, we've got a really important process called crossing over. It concerns the homologous chromosomes. Now, homologous chromosomes have the same genes in the same place. The homologous chromosomes come together in a process called synapsis, and they form a tetrad. The non-sister chromatids overlap each other at the chiasma, plural is chiasmata, and they swap genetic material. The homologous chromosomes become recombined. This results in unique combinations during independent assortment. Okay, if we're looking at meiosis, interphase has occurred. The single-stranded chromosomes have become double-stranded. In prophase 1, the nuclear membrane breaks down, the DNA becomes visible as it condenses, crossing over occurs, and spindle fibers start to be formed by the centrioles. In metaphase 1, the homologous chromosomes attach to the spindle by their centromere. The tetrads line up at the equator. In anaphase 1, recombined homologous chromosomes separate. Each recombined double-stranded chromosome moves to an opposite pole. This is called independent assortment. In telophase 1, the cell splits in two at the cleavage furrow during cytokinesis, and a nuclear membrane forms around the haploid double-stranded chromosomes. In prophase 2, the chromosomes condense further to become shorter and thicker, and the spindle fibers reform with the help of centrioles. During this, the nuclear membrane also breaks down. In metaphase 2, the recombined chromosomes attach to the spindle fibers at the equator. In anaphase 2, the recombined chromosomes split in half, and each recombined chromatid moves to an opposite pole. Again, this is called independent assortment. In telophase 2, the cell splits in two during the process of cytokinesis, and a new nuclear membrane reforms around the DNA. This creates 4 daughter cells, and all the daughter cells are haploid. All of the daughter cells are genetically different to each other and to the parent cell. There is lots of genetic variation created here due to crossing over in prophase 1 and independent assortment in both anaphase 1 and anaphase 2. Meiosis is perfectly suited to a changing environment, as there's always likely to be some combination that suits the environmental conditions. Thank you for paying attention. I hope you've enjoyed it. I hope it's been a benefit to you. I'll let you like and subscribe if you like, and I'll catch you later. Bye now.